morning, everybody. So what we're going to talk about this morning is an aspect of the immune system where I'm not going to talk about what the immune system does to protect us from infection or protect us from cancer or what happens when it goes wrong. I'm going to talk about ways in which we can use the immune system. So we can take components of the immune system and do things with them. Use the immune system for various things. And the two things that I'm going to talk about are here, immunodiagnostics. So where we can use components of the immune system to look for disease and to look for things that are going wrong in the body. Or we can analyze the immune system in the laboratory to see whether somebody's immune system is functioning properly. And then we'll take a short break, just for 5 or 10 minutes. And then I will talk about how we can use components of the immune system to treat diseases. Immunotherapy. So immunodiagnostics, then a very short break, and then immunotherapy. So, if we are going to want to diagnose somebody or to look for cells in the blood from somebody or to look for chemicals in the circulation of a patient, then we may want to use antibodies to do that. Because antibodies, as you know, are very specific molecules. So you can use them to specifically look for a given protein or given sugar or other structures, and they're very useful as reagents. And essentially, if you want to use an antibody in a laboratory, either in a diagnostic laboratory or in a research laboratory, if you want to use an antibody, you have two main choices. You can use polyclonal antibodies, which is a mixture of different antibodies, or you can use monoclonal antibody, which is just a single specificity of antibody. So I'm going to just spend 2 or 3 minutes talking about those two types of antibodies and how you would make, if you wanted to make a polyclonal antibody, or you wanted to make a monoclonal antibody. Let's say you wanted to make an antibody against a hormone. You wanted to develop a test to measure the level of hormone in the circulation of a patient. Then if you were going to use an antibody to measure the hormone, you'd have to make an antibody against that hormone. So how would you do that? And if you're going to make polyclonal, how would you do that? If you're going to make monoclonal antibody, how would you do that? Well, to make polyclonal antibodies, you would simply immunize an animal. You could immunize a human, but in most cases, not in all cases, but in most cases, you would use an animal rather than a human. For example, if you wanted to make an antibody against cancer cells, nobody is going to want to have themselves injected with cancer cells. Certainly not me, okay? So, you would use an animal rather than a human to do that, okay? And many molecules might be toxic, so you wouldn't want to immunize a human with a toxic molecule. But you could immunize an animal with it, as long as it wasn't at a dose that would kill the animal. So you could take, for example, a sheep, and very often you might want to measure in the circulation of a patient their levels of antibody. So, for example, if you had a patient with autoimmune disease, where the body, as well as reacting against foreign pathogens, against things like bacteria and viruses, the body also starts to react against our own body components, autoimmune disease, like type 1 diabetes, like rheumatoid arthritis, multiple sclerosis. These are autoimmune diseases. And in those diseases, patients have autoantibodies. They have antibodies against self-antigen. Now, if you wanted to measure whether the patient had autoantibodies, you need something to allow you to detect the antibody. Okay? And very often you would use an antibody that had been raised against, for example, human IgG. So you might use sheep anti-human IgG, so you could detect in the patient whether they have IgG antibodies against an autoantigen. And I'll show you in a little bit how you would do that. But in terms of making the antibody, you would immunize a sheep with human IgG, and then you would take, after a while, you give 2 or 3 immunizations, and then you would take blood from the sheep, you would allow the blood to clot, you would spin out the red cells and the white cells, and you're left with serum. And that serum would contain sheep anti-human IgG. Sheep antibodies against human IgG, because you immunize with IgG. If you'd used tetanus toxoid, you would have sheep anti-tetanus toxoid. But now you've used human IgG, so you have sheep anti-human IgG. And that will be a mixture of antibodies. There will be several different clones of B cells in the sheep that make antibody to human IgG, not just a single clone. So if this is IgG, this is human IgG, this is a sheep antibody, you have some antibodies against this bit, some against this bit, some against this bit, some against this bit. Polyclonal, many clones making the antibody. So it's a polyclonal anti-serum. And in many diagnostic and therapeutic applications that we'll hear about, people use polyclonal antibodies. But because it is a mixture, sometimes there will be antibodies that maybe are not useful, or maybe would actually be a disadvantage, would not be an advantageous reagent to use, a polyclonal antiserum. So you may want to make antibody where all the antibody is of a single specificity, derived from one clone of B lymphocytes. In other words, a monoclonal antibody. And there are quite a few different ways that you can make monoclonal antibodies. The traditional way of making a monoclonal antibody that has been around since, I think, 1973, something like that, is to make hybridomas. And a hybridoma is where you take 2 cells and you fuse them together. And the 2 cells that you take are a B lymphocyte, which is the cell that makes antibody, and a tumour cell. And the tumour cell that you use is called a myeloma, and a myeloma is actually a tumour of a B cell. So you take a normal B cell from an animal that you have immunized. You might take a mouse, you might immunize a mouse, so you could immunize a mouse with human IgG. That is foreign to the mouse, so it will make an immune response to the foreign protein, the human IgG. It's different to mouse IgG. So the mouse sees it as foreign, and its B cells, some of the B cells in the mouse will make anti-human IgG. You take those B cells. If you take B cells and put them in culture, you grow them in the laboratory, after about 1 week they die. They're not immortal. They have a short lifespan outside of the body. But you want to keep those B cells alive so that they will secrete the antibody, the mouse anti-human IgG. Tumour cells are immortal. They live and grow forever. Okay? This is why they are tumour cells. Unlike normal cells, they just keep dividing, they expand up in number. That's the problem with a tumour. Keeps dividing, keeps growing. It will spread throughout the body eventually. So you take a tumour cell, because the tumour cell has the property of immortality. It is immortal. So you take a cell that makes the antibody you want, but is not immortal, and you take a cell that is immortal, and you fuse them. And then in that cell you have the gene that encodes the antibody you want, but also genes that make the cell grow forever. And this is the way that you do it. So you would immunize a mouse with an antigen, and that antigen has different parts of it. Just like this bottle has a bit here, bit here, bit here, bit here. The antigen has different parts. So on the antigen you have A, B, different areas on the antigen. And when you immunise a mouse or any other animal with that antigen, there'll be some B cells in that mouse that will make antibodies to the part A of the antigen, some to part B of the antigen, etc. So you have anti-A, anti-B, etc. Now you want to take B cells from that mouse. You could take blood, but there are not huge numbers of B cells in the blood. There are quite a few. But there are more B cells in the spleen. It's very easy to take the spleen. So you take the spleen out of the mouse, you mash it up, and you get cells. Most of those cells will be B cells. There'll also be T cells there and some dendritic cells and other types of cells, but there'll be lots and lots of B cells. You then take those B cells, and you'll have some B cells that are going to make antibody to part A of the antigen, some that make antibody to part B of the antigen, and so forth. And you fuse these cells with an immortal B cell tumour, which is called a myeloma. So you fuse them. And it's very easy to do that. When this technique was first developed by Kohler and Milstein, who won a Nobel Prize for developing this technique. They worked in Cambridge in the United Kingdom. They developed this technique, won a Nobel Prize for it. When they first did this, it's actually interesting because when they first did this, they were not trying to make monoclonal antibodies. The idea was not to make monoclonal antibodies. The idea was they wanted to study B lymphocytes. But when they tried to grow B lymphocytes in the laboratory, they would always die. So the aim was to make B cells grow forever. They didn't want to make monoclonal antibodies, didn't think about making monoclonal antibodies. But then they did think about making monoclonal. They realized that they could use it to make monoclonal antibodies, but that was not the aim. And they used a virus to fuse the B cell and the myeloma cell. They used a virus called a Sendai virus. Okay? But they also realized that there were easier ways of making the cells fuse. And now we use a chemical called polyethylene glycol, PEG. And when you add polyethylene glycol to cells, it makes them fuse. The membranes fuse, and you get hybrids. So you get a hybrid cell, and these hybridomas, because they're hybrid cells, they're called hybridomas, they are immortal. Now, you have a test tube and you have a mixture of cells. So you have B cells that are fused with myeloma cells, but you have a mixture. So now you need to separate out the individual cells. And you do that by using a technique called limiting dilution. And what you do is you take the hybrids and you plate them out into a plate that has 96 wells. It's called a microtile plate. And what you do is you dilute the cells so that when you put them in the wells, there are 60 cells that go into 96 wells. Now you can't have 2/3 of a cell. You can either have no cells or 1 cell or 2 cells, but you can't have a part of a cell. Okay? But you take 60 and put them into 96 wells. Some wells will have no cells, some will have 1, some will have 2, maybe some occasionally will have 3 cells. Okay? But on average, there is 2/3 of a cell per well. If you do that, then some of the wells, no cells will grow because there weren't any cells there. Okay? But where there were cells, the cells will grow and they'll divide because they've been made immortal and they will start dividing. You then take the wells that have some cells in, you just look down the microscope and you can see the cells. You take wells where there's some cells, and then you dilute those out again. And you put 60 cells into 96 wells. So you do it twice. And this is called limiting dilution. And if you do that twice, and some cells grow in a well, then statistically it is very, very likely that that was derived from 1 cell. And you can go on and analyze it and show that it is indeed a single clone of cells, monoclone. So then you'll have some clones that are making antibody against part A of the antigen, some clones against part B, and so on. And then you can grow those up to a very large volume, and they will secrete a very large amount of monoclonal antibody. Now this is the traditional technique I think it's 1973 it was developed, I can't remember, but around about then. So it's been around for a few decades. More recently, other techniques have been developed to make monoclonal antibodies, but this is still the way in which most monoclonal antibodies are made today. But the newer techniques are useful and are used, and quite a few monoclonal antibodies, particularly ones that are used for immunotherapy, quite a few are made using recombinant DNA technology as a newer approach. And one of the ways, there are several ways you can do this using recombinant DNA technology, but one way that is being developed over the last few years, particularly at the Scripps Institute in La Jolla in California in the USA, and also in Cambridge in the UK, two main groups that developed this technology. Now lots of other people are using it. It's called bacteriophage display, and I'm just going to spend a few seconds telling you how you do this. So you take B cells from a mouse that you've immunized, or you can take human B cells just from somebody that isn't immunized, and you isolate the messenger RNA, and then you isolate messenger RNA that is encoding IgG antibody, for example. You make a complementary DNA copy from the messenger RNA, so you have double-stranded nucleic acid. And then you use the polymerase chain reaction to amplify off the nucleic acid. So you have lots of nucleic acid with a sequence that encodes antibody. You then put this antibody into a sequence that is encoding a bacteriophage. A bacteriophage is a virus that infects bacteria. So you then have a sequence that is like this. And what you've got there is a leader sequence. The L stands for leader, which is the first part of a sequence that will encode for a protein and allow it to be secreted. Then the variable region from the light chain and the constant region from the light chain of the antibody. And then you have a stop codon. So that will encode an antibody light chain. And then you have another sequence after the stop codon that will encode the antibody heavy chain. So you have leader sequence, you have variable of the heavy chain, and the first constant region of the heavy chain. Okay. So this would encode what you would call a Fab fragment, which is just, if you have an antibody like this, the Fab fragment is just this bit. Okay? It's the light chain and the first part of the heavy chain, but it contains all the variable region. It contains the variable region for the light chain, variable region for the heavy chain. But the clever thing about this technique is that next to that, here you see something called P3. And P3 is a coat protein from the outside of a bacteriophage. So you produce a fusion protein. Part of the protein is an antibody heavy chain, and part of the protein is the coat of a bacteriophage virus. You then have a situation where you have a bacteriophage that produces its coat protein, which is called P3, but fused onto that coat protein is the Fab fragment of an antibody, one arm of the antibody. Now what is really clever here is that within the phage you have the gene. So you have a little packet that contains the genes you're interested in, and on the outside you have the antibody. Now what you can do is use a technique called panning, where you have the antigen you're interested in. Let's say this is the antigen you're interested in. Okay? And it's here. And there are lots and lots of other antigens that maybe you're interested in, but you put onto a plate the antigen you're interested in. If the antibody doesn't bind, then you can wash the antibody away. But if the antibody binds, then it will bind to the antigen on the plate. So you can have millions and millions and millions and millions and millions of these phages. You can have like 10 to the 15, 10 to the power of 15, a huge number of phages, each with a different antibody gene inside. So imagine this water is the gene for the antibody, and this is the bacteriophage, and then imagine this is the antibody here. Okay? So you have a bacteriophage, inside you have the gene, here is the antibody. The antibody recognises the antigen, it will bind to a plate on which you put the antigen. You then wash away anything that isn't bound, and then you can elute this, you can change the pH for example, you can elute this, you can break it open, and you've got the gene that you want for this antibody. Okay? And the other pages will have genes for other antibodies. This makes it a very, very powerful technique because you can select out of millions and millions and millions and millions and millions of different antibodies just the one you want. Okay? And perhaps I'll just mention, I haven't got a slide to show this, but I'll mention that another way that is being used to develop human monoclonal antibodies is to make mice that are transgenic for human antibody genes. So you can make transgenic mice for any gene, so you can put a human gene or any other gene into the mouse to make what's called a transgenic mouse. So the mouse has the mouse genome, all the normal mouse genes, plus a human gene. And you can take human antibody genes and put them into mice and make transgenic mice from which you can make monoclonal antibodies. And that's another technique that is used to derive human monoclonal antibodies. So having made antibodies, whether they are polyclonal antibodies or monoclonal antibodies, what can you then use them for? You can use them in diagnostic assays to test for things in patients, whether they have raised hormone levels, whether they have autoantibodies, whatever you want to test. Whatever the specificity of the antibody is, it allows you to test for that. You can use them in therapeutics, or you can use them in the laboratory as research reagents. Anybody working in a laboratory, if it's an immunology laboratory, they will be using antibodies, almost certainly, at some point in their research. But in other areas of biology, people also use antibodies. People who are not immunologists at all, neuroscientists, embryologists, endocrinologists, they will use antibodies as tools. They're not interested in the immunology, but they're interested in having a reagent that is specific for molecules they are interested in. So we're not going to talk about the use of experimental antibodies as experimental tools this morning, but we are going to talk about the use of antibodies for diagnostics and for therapeutics. Now, if this was an antibody, you'd be able to see it if it was this. But you can't see antibodies. Antibodies are quite small. Okay? An antibody heavy chain is 50,000 Daltons. An antibody light chain is 25,000 Daltons. An antibody has 2 heavy chains, 2 light chains. 50 plus 50 plus 25 plus 25. It's 150,000 Daltons. It's too small. If an antibody was on this table, you couldn't see it. If you've got a microscope, you put the antibody under the microscope, you still couldn't see it. It's too small. If you use an electron microscope, you can just about see an antibody. Under an electron microscope, you can see antibodies, but only just, and it's not a technique that you could easily use to measure antibodies. So you need to develop methods to allow you to be able to see antibodies when they're too small for your eyes to see. And there are many, many different methods that you can use, but most of them rely on putting something onto an antibody that allows you to measure it. So you tag or you label the antibody with something. And one of the most commonly used techniques is this technique called ELISA, Enzyme-Linked Immunosorbent Assay, ELISA. And in this assay, you label an antibody or tag an antibody with an enzyme. But not any old enzyme. It needs to be an enzyme that causes a colour change in a substrate. All enzymes work on a substrate. They change something in some way. That's what an enzyme does. And the enzyme you use in the ELISA assay has the property that it changes the colour of a substrate. And there are several different enzymes that are used, and they will change different substrates and make different colours. And you could use this to measure the binding of an antibody to an antigen, or the binding of one antibody to another. So you could use it to measure the binding of sheep anti-human IgG to human IgG, for example. Or you could use it to measure the binding of anti-tetanus antibodies to tetanus. And the way that you do this is that you coat plastic plates, for example microtiter plates, which is what you've got here. This is the same kind of plates as you do the hypodermic dilutions in. Okay? 96 wells, 12 by 8 wells. You can do an ELISA by coating the wells with the antigen you are interested in. And it is very fortunate and very easy because proteins stick to plastic very, very strongly. If you put a protein in a solution onto a plastic well or plastic dish, the protein binds very, very strongly. You don't need to chemically link it, it just binds. Okay? It's not completely permanent binding. It's not covalent binding. Eventually, it will come away, but over a very long period of time. Basically, it's stuck there very, very hard. So you coat the plastic wells with the antigen you're interested in. So that is at the very top. Okay? You then wash away any antigen that hasn't bound. You then add the antibody you're interested in. So, for example, maybe you want to measure autoantibodies. And maybe you have a patient with a disease called, there's an autoimmune disease called SLE, systemic lupus erythematosus. It's quite a long name. But in these patients, they have autoantibodies to DNA and to other nuclear antigens. Okay? So you could put DNA on the plate and say, does the patient have antibodies that bind to DNA? So you could coat the plate with DNA. It's a bit more tricky than a protein. You have to link the DNA to the plastic, but you can do that. And then you would wash away anything that hasn't bound, and then you would come in with antibody. If the patient has autoantibodies to DNA, it will bind to the antigen. If the patient doesn't, they won't bind. So you wash away anything that isn't bound, and you're left with antibodies stuck onto the antigen. But you can't see the antibody. So now you need to have some way to see whether there's antibody there. And one way would be to add sheep anti-human IgG that has an enzyme coupled onto it. So that would be this bit here, okay? Where I have enzyme portion and ligand. That could be a sheep anti-human IgG with a particular enzyme. An enzyme that is often used in ELISAs is called alkaline phosphatase. So you have sheep anti-human IgG coupled with alkaline phosphatase. If there's antibody bound, then that will bind. If there isn't antibody bound, then it won't bind. You can wash, and then you add the substrate, which is called a chromogen. And if there was patient antibody, the ligand will bind, the sheep anti-human IgG will bind, the enzyme will be there, it will cause a colour change in the substrate. And this is what you would see. And in any assay, it is very, very important always to have a positive control, so you know the assay works, and a negative control, to also show that the assay works properly. And in these wells here, in this last column, is the negative control. And in this column is the positive control. So we can see it's worked properly. Where it should be positive, it's positive. Where it should be negative, it's negative. And then, what we have along here are dilutions of the patient's serum. So the patient's serum is diluted 1 in 2, 1 in 4, 1 in 8, doubling dilutions. And then the patient serum is added to the wells that are coated with the antigen. Let's say it's DNA, and we're looking for anti-DNA antibodies. And you can see this patient is positive. When the antibodies are diluted, 1 in 2 is positive, 1 in 4 is positive, 1 in 8 is positive, 1 in 16 is positive, 1 in 32 very weakly positive. We'd say that this is the endpoint dilution where it's positive. We'd say the titer of antibody, which gives you an idea of the amount and the strength of binding. The titer in this patient is probably about 1 in 16, because this is quite weak. So we'd say that's 1 in 16. Here, the titer is only 1 in 2, because there's very little binding there. This patient has stronger antibodies, probably 1 in 64. This is stronger. You can dilute it to 1 in 128, and it's still positive. This patient is negative, doesn't have any autoantibodies. Okay. So you can use ELISA to detect whether a patient has autoantibodies, and you can see how much autoantibody, a little bit or a lot. The more you can dilute it, the more more antibody is there. Okay. So this is looking for autoantibodies, but you can use this technique to look for hormone levels. You could have antibody against hormone on the plate, and then you add serum to measure the hormone level, and then you use another antibody to a different epitope, a different part of the hormone, and see whether there's hormone there. So there's many, many different ways you can use an ELISA technique. Another immunodiagnostic technique that is very often used is to use immunofluorescence. In this technique, instead of labeling an antibody with an enzyme, you label it with a dye that emits fluorescence when you shine light of a particular wavelength onto the dye. And very often, you will use a dye that fluoresces when you shine ultraviolet light onto it. And this technique, unlike the other techniques, I mean, ELISA you can also do in various different ways. What it shows on this slide is 2 different ways of doing immunofluorescence. This is a direct technique, and over there we have an indirect technique. So in the direct technique, you could do this in a well with things coated with the antigen, or you could do it on a section of tissue, or you could do it on cells. Okay. So here we have a tissue section. So let's say you wanted to measure antibodies against, let's say still a patient with SLE, a patient with systemic lupus erythematosus that has autoantibodies against DNA. If you cut a tissue section, then there'll be nuclei in the tissue, and there'll be DNA in the nucleus of the tissue. So you can see whether the patient's antibodies bind to the antigen. And you could label the antibodies from the patient with the dye, and then see whether the patient's antibodies bind. That would be a direct immunofluorescence test. However, if you were going to measure antibodies in a patient, it's much better to use an indirect test, because then you don't need to label each antibody from each patient. What you can do is use a reagent that you can buy that is labelled that recognises human IgG. So you could buy sheep anti-human IgG. You don't have to catch a sheep and immunise it yourself. You can buy sheep anti-human IgG, and you can buy it labelled with an enzyme, labelled with fluorescein, a fluorescent dye, or many other types of label. You just buy it from the company. So then you have the tissue section, you add the patient's antibody, you leave it for a while, usually about 30 minutes. You then wash away, and if there are no autoantibodies there, all the antibodies will be washed away. If there is autoantibody, it will have bound. And then you detect the antibody by adding a fluorescinated, this is fluorescein, dye, FITC, fluorescein isothiocyanate. This is fluorescinated anti-IgG, it will bind. You then wash. If there's no antibody here, none of this will bind. If there's antibody here, some of this will bind. And then you can shine ultraviolet light onto the tissue section, and you can look for fluorescence. And this is what you would see. So this is a section, these are both tissue sections of normal thyroid. Okay? Thyroid is an organ, an endocrine gland in the neck that makes thyroid hormone. And thin sections of normal thyroid have been cut, put onto a glass slide, and then this immunofluorescence test has been carried out. So the section of thyroid has been incubated with serum from the patient, then washed, and then incubated with fluorescinated anti-human IgG. And what you can see, ignore the colour, the difference in the colour, but what you can see is that the staining pattern is different in this patient and this patient. Because this patient here is a patient with systemic lupus erythematosus, SLE, which I mentioned these patients have autoantibodies against DNA and other nuclear antigens as well. Sometimes say they have anti-nuclear antibodies, ANA. And what is staining here are the nuclei in the thyroid cells. If you look here, you can see that this patient is also positive. This patient also has antibodies against thyroid. But the pattern of staining is different. The nuclei are not staining. These black holes are the nuclei, and they are not staining. But the cytoplasm of the cell is staining. Particularly the surface of the cell is staining strongly. This is a patient with a different type of autoimmune disease. This is a patient with an autoimmune disease against the thyroid gland itself, a disease called Hashimoto's thyroiditis. And these autoantibodies are not recognizing DNA because the nuclei are not staining. What these autoantibodies recognize is something called thyroid peroxidase, which you find in the thyroid, that is involved in making thyroid hormone. If you did this test again, using the same sera from the same two patients, but instead of using thyroid, you use liver, or lung, or brain, or other tissues, this patient would be negative. There'd be no staining, because the antigen that is recognized is only found in the thyroid gland. Thyroid peroxidase, the only place you find thyroid peroxidase is in the thyroid. It has a specific job to do. It helps in making thyroid hormone. You only make thyroid hormone in the thyroid, nowhere else. But if you were to use this patient's sera on lung, liver, brain, any other tissue, it would still be positive because it's recognizing DNA in nuclei. And all tissues have cells that have nuclei that have DNA. So this disease, systemic lupus erythematosus, is an example of an autoimmune disease that is systemic. That's why it's called systemic, throughout the body. It's a non-organ specific autoimmune disease. Whereas this autoimmune disease is organ specific, specific for thyroid. This patient has thyroid autoimmune disease, a particular type called Hashimoto's thyroiditis. So you can use immunofluorescence diagnostically to distinguish different types of patients. And you can say, this patient has SLE because they have antibodies against nuclei. This patient has thyroid autoimmune disease, a different type of autoimmune disease, because it has antibodies that bind to a different antigen. Now, as well as doing immunofluorescence on tissue sections, very often in a diagnostic laboratory or in a research laboratory, immunofluorescence is carried out on single cells. So, for example, if you had a patient that was infected with HIV, human immunodeficiency virus, usually when one becomes infected with HIV, eventually, after a period of time, it might be months or years, the patients go on to develop AIDS, acquired immune deficiency syndrome. It takes a while after infection to develop AIDS, sometimes it can be years, but most patients eventually will develop AIDS. And one of the characteristics of AIDS is that in the blood, the lymphocytes change because the HIV virus infects helper T cells. And helper T cells have on their surface a molecule called CD4. And the virus, HIV, enters cells by binding to CD4. And eventually those cells become destroyed. The other major sort of T lymphocyte in the blood has CD8 on the surface. So we have helper T cells that have CD4 on the surface and cytotoxic T cells that have CD8 on the surface. And in a normal individual, there are more CD4 cells in the blood than CD8 cells. But in AIDS, because the CD4 cells become infected with virus and become destroyed, eventually those patients lose CD4 cells and they end up with having more CD8 cells than CD4 cells. So most people have more CD4 cells than CD8, but in patients with AIDS they have more CD8 than CD4. And you can use fluorescinated antibodies to detect whether cells in the blood have CD4 on them or CD8 on them. So this is a cytotoxic T cell. It's a T cell, so it has a T cell receptor. All T cells have a T cell receptor on it. So this is a cytotoxic T cell. It has a T cell receptor, but it also has CD8 on its surface. This is a helper T cell. This also has a T cell receptor, because all T cells have a T cell receptor. But this does not have CD8 on its surface. It has CD4, which most helper T cells have. So you can use an antibody, a monoclonal antibody, that is specific for CD8. And this monoclonal antibody can be labeled with a fluorescent dye. Then you can incubate these cells with that antibody. You incubate blood, lymphocytes with this antibody, and any cells that have CD8 will bind the antibody and will be fluorescent, but cells that have CD4, they don't have CD8, so there will be no antibody binding. Then you could use another monoclonal with a different dye, perhaps a red dye rather than a green dye, and you could have an antibody against CD4 that had a red dye on it. And the red dye would bind to these cells, but not to these cells. Now what you could then do is look down the microscope that is illuminated with ultraviolet light, and you could see the cells that had CD8 on them. So this is a CD8 positive cell. Has CD8 on its surface. Can anybody see the CD4 positive cells? Maybe one here, maybe one here, maybe there's one there. Okay? So the CD4 positive cells would not stay. Okay? If they'd been incubated with an antibody that was against CD4 with a red dye, then we might see some red cells here. Okay? But because they've only been incubated with a single antibody, and that is against CD8, you can't see any other cells. But there's some there that you can't see them. Now, this is how these techniques were originally done. When I was your age, I would stare down a microscope and I would count hundreds and hundreds of cells. And it's very tedious. It takes a long time. And if you have lots and lots of samples, you kind of go a bit crazy because you're sort of looking at lots and lots of slides with cells on, and it's not very easy to do. So nowadays, rather than look down a microscope, people use a machine that does it for them. And the machine is called a flow cytometer. And the very good thing about a flow cytometer is that you can analyze a very high number of cells, and therefore you get a much more accurate measure of the number of positive cells. If you count 200 cells under a microscope, maybe you count another 200, you'll get a slightly different percentage. First time you may say 7%, okay, and then you count another 200 cells and you may get a value of 9%. Count another 200, you may get a value of 5%, okay. You'd have to count thousands of cells to get an accurate value, and you looking down a microscope, you just can't do that. It just takes too long to do it, okay. But this machine will analyze, this flow cytometer will analyze 100,000 cells or more in less than 1 minute. So gives a very accurate result, a very rapid result. Not only can you measure fluorescence, but the flow cytometer also will give you information about cell size, and it will give you information about how granular the cytoplasm is in the cell. So it gives you other properties of the cell, physical properties of the cell. You can analyze whatever you're probing for with fluorescent antibodies. As long as you have an antibody to a molecule that is present on the surface of the cell, then you can have an antibody against T cell receptor, an antibody against CD4, an antibody against CD8, an antibody against CD1, CD2, CD3, CD5, CD250, whatever, okay? If you have a monoclonal antibody, then you can measure that particular property of the cell. And this is used as a routine kind of analysis to look for CD4 and CD8 numbers in patients with AIDS. Also to look for the expansion of particular clones of cells in leukemias and in lymphomas. And you can also modify the technique to look for things inside of cells. Now, antibodies, they're fantastic molecules, but they can't get inside cells. Okay? A living cell excludes antibodies. Sometimes they can be taken up into a vacuole, but they don't get into the cytoplasm of cells in a natural situation. But when you use this technique, you can take cells and you can fix them with a fixative, so that they're not living anymore, but they're still, you can still analyze them. You can fix them with a fixative, and you can permeabilize them. You can incubate them with a chemical that puts pores into the cell membrane, and then antibodies can get into the cell. So you can use them to look at cytokines inside cells, which are chemical messengers that cells produce. You can use them to, you can use it to look at apoptosis. I'll show you that in a second. For that, you can use live cells. You don't need to use dead cells. The cells undergoing apoptosis are dying, because that is what apoptosis is. So you can modify it in this technique in various ways to analyze a lot of different things. When the flow cytometer analyzes cells, the information is then fed into a computer, and software in the computer generates various types of images so that you can analyze what is going on. You can look with your eye and see what is going on. And there are various ways in which the information can be presented by the software in the computer. And this is a dot plot, which is a way that very often flow cytometry data is visualized. So what we have here is CD4. And along this axis, and you can see little dots here. Each dot represents a cell, and further along this axis is CD4 positive. So cells here do not have CD4, but cells here have CD4. Okay? The further along, the more CD4 there is. Okay? Now, what you tell the computer to do is, at a certain level, you say, anything above this level, I'm going to say is CD4 positive, and anything below that level, I'm going to say is CD4 negative. And you tell it to draw a line. So you tell it to draw a line here, and you say, this is so weak, this binding, there's some binding, but it's so weak that it's just a background binding, and we're going to say these cells are CD4 negative. Whereas anything above this is CD4 positive. And then on this axis, they've used another monoclonal antibody with a different dye. So let's say this was fluorescinated antibody against CD4. Maybe that is a rhodaminated, which is a red dye, antibody against CD8. And the same thing, anything here is negative, anything at the top is positive. And they've told you to draw a line, a horizontal line there. Okay. So now you have 4 squares, 4 quadrants. Anything here is CD4 negative, CD8 negative. It's negative for both. Anything at the top there is CD4 negative, but CD8 positive. Okay? This is CD4 positive, CD8 negative, and at the top is double positive, CD4 positive and CD8 positive. Now these cells, if you look at the very, very top, are cells that are called thymocytes. And thymocytes are T cells that are developing in the thymus. Okay? The thymus, the thyroid is here, the thymus is there, okay, in the thoracic cavity. The thymus is where T cells develop. And the only place you find T cells that express both CD4 and CD8 is in the thymus. Except in some rare leukemias, the only place you ever find CD4 positive and CD8 positive cells, a cell expressing, single cell expressing both CD4 and CD8, is in the thymus, in developing T cells. Because once those T cells become mature and are ready to leave the thymus, they switch off expression of either the CD4 gene or the CD8 gene. And they just express CD4 and not CD8, or just CD8 and not CD4. And sometimes we refer to the T cells in the thymus as being double positive. They are positive for CD4 and CD8. And then they become single positive, positive only for CD4 or CD8, but not both. So you can see in the thymus, in the top right on the top thing, lots of positive cells that are positive for CD4 and CD8. Whereas if you look in the spleen, which is here, splenocyte cells in the spleen, then in the spleen you get lots of B cells, as I mentioned, because you can make hybridomas from spleen B cells. But you also have T cells in the spleen as well. But these T cells will either be CD4 negative, CD8 positive, or there'll be some double negatives, or there'll be CD4 positive but CD8 negative. So these will be T cells that are CD4 positive T cells. These will be T cells that are CD8 positive T cells. And these will be other types of cells, B cells for example, which don't have CD4 or CD8. Okay? So if you take splenocytes, which are spleen lymphocytes, many of them are B cells. These are the B cells. They don't have CD4 or CD8. These are helper T cells, which are CD4 positive. These are cytotoxic T cells, which are CD8 positive. And hardly any, this is kind of the background here, hardly any double positives. In an ideal situation you would have nothing here, but there are a few double positives, but those are just kind of probably just noise in the systems in the background. So you can use flow cytometry to analyse cells like that. Now, before we take a short break, we're going to take a very short break before we talk about immunodiagnostics. I also want to tell you how you can use flow cytometry to measure apoptosis, programmed cell death. Because in many situations, especially in the laboratory, experimentally, you want to measure whether cells are undergoing apoptosis. And there are many different ways of measuring apoptosis, but a very simple and quick way is to use flow cytometry. And this relies on the fact that in a normal cell, you have a cell membrane, a lipid bilayer, and on the cytoplasmic, the inside part of the lipid bilayer, you have a molecule called phosphatidylserine. And phosphatidylserine is only found in a living, viable, normal cell on the inside of the membrane. Okay. Now, I've mentioned that antibodies can't get inside cells, and that's true of other molecules as well. Many proteins and other types of molecules can't readily get inside a cell. There needs to be a specific transport system. Things are taken into a cell, but there's a specific transport system. Okay? And you can use a molecule that normally does not go inside cells, that is called annexin. And annexin binds phosphatidylserine. So just like an antibody binds an antigen, so annexin binds phosphatidylserine. And you can label an antibody with a fluorescent dye. For example, FITC, F-I-T-C, fluorescein isothiocyanate. It's the dye that was green fluorescence that you saw. So you label annexin, a particular type of annexin called annexin 5. You label annexin 5 with fluorescein isothiocyanate. You have a conjugate of FITC and annexin 5. If you incubate that with normal viable cells, it doesn't bind, because what it recognizes is phosphatidylserine, and that's on the inside of the cell membrane. It's not accessible. But when cells undergo apoptosis, a number of things happen. The DNA starts to be degraded, but also the cell membrane starts to flip. Bits of the cell membrane that were on the inside come on the outside. And that means that phosphatidylserine, normally only on the inside, starts to appear on the outside of the cell. And that's a very early indicator of apoptosis. When cells are just beginning to undergo apoptosis, bits of the membrane that were on the inside become on the outside. And then the annexin can bind, and you can look down the microscope, or you can use flow cytometry to measure that. And you could probably just about see, because of the light, maybe a little bit difficult, but you can just about see green on the outside of this. People see that? Yeah? So that is phosphatidylserine on the outside of a cell undergoing apoptosis. And maybe you can see this. This is a bit later on in apoptosis, and now more of the membrane is on the outside. That was on the inside is now on the outside, and the fluorescence is getting stronger. But as well as looking down the microscope, you can also use flow cytometry. And you can show it like this, in this kind of analysis, which is a dot plot. Or you can tell the software in the computer, you can say, show me it as a dot plot, or you just press a button, show it me as a histogram. Okay? And this is a histogram. So what you have here is fluorescence intensity going this way, number of cells going this way. So there are lots of cells here that are not fluorescent, because this is a group of cells that have not yet undergone apoptosis. You can then incubate those cells with a chemical that causes apoptosis, and then the annexin 5 will bind, and you'll get a peak here. So this is another way of visualizing data generated by flow cytometry. So these would be negative cells, these would be positive cells. And you could do the same when I was showing you the CD4 and CD8. You could tell the computer to show it to you in this way rather than the dot plot way. Okay, and then just before we come on to talk about immunotherapy, I just want to finish off very briefly talking about immunodiagnosis of cancer. And this you can use to monitor tumours. So you could look for tumour antigens in the blood. You could look for the development of metastases. This is when the main tumour, cells leave the main tumour and spread throughout the body. So you can look for micrometastases, very small amounts of metastases, which would be a bad sign in cancer that the cancer is spreading. So you'd maybe want to treat that patient very quickly before the cancer gets a chance to spread anymore. You can use antibodies to look where the tumour is, to image the tumour. And again, you can use techniques like we've already mentioned on cells or tissues. You have a tumour antigen, you can use a monoclonal antibody, and then you can use a secondary antibody. And you can use a technique that is essentially it's the same as an ELISA technique, but there's one difference, and that is that the substrate that you use in an ELISA technique, the substrate is soluble, and you can see the colour in the well, like you saw the blue colour in the well of the ELISA plate. Okay, that's a soluble product from the substrate. In this particular technique that is called immunohistochemistry, then you use a substrate that is not soluble, and it precipitates on the tissue section. So you get a colour change, but the substrate stays on the section of tissue, or on the cells that you put onto a glass. And here you have an antibody against something called cytokeratin, and cytokeratin is very commonly expressed in cancer cells. So this would indicate that this cell was a cancer cell. And then the doctor would need to start treating this patient because he can tell that they have cancer. So this is from a lymph node, and it's showing that the cancer cells have spread from the main tumour into a lymph node. So the surgeon has taken the lymph node out, there's a biopsy, and asked, has the cancer spread? Maybe the patient had breast cancer, and maybe it spread to the local lymph node. And you can tell that from this diagnostic test. There are a large number of different tumour antigens, carcinoembryonic antigen, CEA, in colorectal cancer, prostate-specific antigen in prostate cancer, molecule called alpha-fetoprotein in liver cancer, various mucins, which are carbohydrate antigens, CA125, 19-9 in ovarian cancer. So you can use immunoassay to detect these cancer antigens, and that gives you an indication that cancer is present, and then the doctor can treat it. But one has to be cautious because not all tumour-associated antigens necessarily mean that there is a cancer there. It's indicative, but it doesn't for sure say that there's a cancer there. You also need to find the cancer cells as well as finding these ones. What it is very useful for is measuring in a patient that has already been diagnosed with cancer and treated whether there's a recurrence of the cancer. So this is a particular cancer-associated antigen, carcinoembryonic antigen, which I mentioned on the previous slide, CEA. It's associated with colorectal cancer. So this would be a patient, the tumour has been removed by surgery, everything seems to be fine, there's no CEA in the circulation of this patient, so there's no evidence that there's any residual cancer. The surgeon has removed all the cancer, there doesn't seem to be any cancer left. But you can monitor the problem. But then, after a period of time, you can see that you can begin to measure a level of CEA that is more than the normal level. So now the surgeon or the doctor could start treating this patient again very quickly. There's a recurrence of the cancer is coming back, treat the patient very quickly. So we can use these immunodiagnostic and immunomonitoring techniques to measure cancer antigens to detect very early on that the cancer is coming back. You can also find out where the cancer is by using antibodies that have a label. So, for example, you could label antibody against CEA with an isotope of iodine called I131. This is an X-ray radiograph of the chest, and you can see bits of cancer that have spread to the lung. Okay? But if you use a technique called immunoscintigraphy, if you use this, so you inject a patient with I131 labeled antibody against CEA, and then you scan them with a gamma camera, you can see where the radioactivity is. So you can see much more clearly than you can on an X-ray that this cancer has actually spread to many, many places in this patient. So everywhere you can see here, there's cancer, and it's spread throughout the patient. This is by looking for an antibody to a cancer antigen. Where does that go to in the patient when you inject it? It will only find where there is cancer. Okay, so in summary, antibodies can be used in many applications, including diagnostics, therapeutics, and also as research tools. So I'm going to take a short break now, and then I'll come back and tell you a little bit about immunotherapy, how you can use antibodies to treat various kinds of clinical conditions. So shall we say we meet again at 9:10? So I'm just going to give you 7 minutes break, okay? 9:10 we'll start again.